welcome to Science Friday.
Here's some for E as well. The one on the top here is one of the ways that you can define E.
What makes a field more likely to have an unbalanced gender distribution?
So this can be, like, in the case of this paper, gyrification or brain folds.
A neuroscientist started to look at the brain as
like the
more as an interconnected system.
It has incredibly negative consequences.
Thank you, guys. I'm Thomas. I'm a junior here, and I'll be presenting on microfluidic-assisted FISH analysis of breast cancer. So before we jump into anything, I want to clarify what microfluidics are, because I've talked with some people and there's a varying degree of understanding. So microfluidics is a complicated field of nanofabrication that spans a bunch of different disciplines, but it really boils down to the simple idea of fabricating tiny tubes to control small volumes of liquids. That's really it. Why do we care about small volumes of liquids? Well, at the nanoscale, fluids behave in very different ways than we're used to in the macroscopic world. And researchers are able to take advantage of these unique properties to do some really interesting work. And so microfluidics is the discipline that kind of tames these microscales of fluids to do research. So you hear me talking about kind of throughout the presentation these microfluidic chips. A microfluidic chip is just how researchers actually build these nanotubes and these nano devices. So your most common kind of microfluidic chip is made out of a PDMS silicone resin. And these lines here are actually those tiny tubes where the fluid flows through. And this right here is just a simple mixing microfluidic chip. The pattern can be in whatever layout you need. But this basically allows you to manipulate those super small volumes of liquids. Other kinds of microfluidic chips include this one, which is actually made out of paper and wax, and this one, which is 3D printed out of glass. So the idea is really, microfluidics is a very broad topic and subject and used all over the place, but it really just boils down to the idea of controlling very small volumes of liquid. So with that, why combine microfluidics and cancer? Well, microfluidics is this really exciting new field which has emerged in the last decade or two. Every month there's incredible new research that pushes technology further. And it's kind of hot and exciting, and every university seems to have a microfluidics lab. On the other hand, the FISH procedure, which I'll be talking about in a minute, is not really the same. It was popularized in the 1980s, and since then hasn't really had much progress or work done. And so the idea that the researchers had were to take this new and exciting field and apply it and revitalize this somewhat old and belated test. So why breast cancer specifically? Well, both my grandmother and aunt had breast cancer, and so I've grown up around the disease and have seen what it's done to people, and it's really challenging. And so, while this paper certainly does not solve breast cancer, what it does do is lower the barrier of entry for someone who's diagnosed with breast cancer and helps them begin their process of determining which chemotherapy is right for them and overall just trying to aid in their prognosis. So, in about 15 to 20% of invasive breast cancer cases, patients have what's known as Her2 overexpression. Her2 is a gene, the human epidermal growth factor 2, and in a Her2 overexpressing cell, that Her2 gene is literally overexpressed. So you find multiple copies of it in the cancerous cell's DNA. Now, a Her2 positive or Her2 overexpressing cell typically means that the cancer is going to be more aggressive. However, that Her2 overexpressing cell also has a very unique feature, the fact that it has all these Her2 genomes. And researchers and doctors are able to use that fact that these cells have all these Her2 genes to develop very specific drugs, namely trastuzumab and others, which is a kind of chemotherapy, which target specific cells with this Her2 overexpression. So it means that even though you're diagnosed with Her2 overexpressing cancer, which is generally more aggressive, you're able to take these drugs which specifically target that and actually result in a better prognosis for you. So this means that if you are diagnosed with breast cancer, it's imperative that you get tested for Her2 overexpression. And if you are one of those 15 to 20% who have Her2 overexpression, then you can take these very specific chemotherapies tailored to your own situation, which help you in the treatment and recovery. So one way doctors actually detect this HER2 overexpression is through FISH, or fluorescence in situ hybridization. So FISH starts with your target DNA. In this case, it's going to be your cancerous tissue sample from the patient. And then using heat, you denature the DNA, in effect splitting it apart. Then you introduce what's known as a FISH probe. And a FISH probe right here has two different sections. The first is a fluorophore, and the second is a complementary strand of DNA. Now these little fluorophores here glow when exposed to a certain wavelength of light. So you might be seeing where this is going. By combining these fluorophores with that complementary DNA strand, which is specific for the HER2 gene, you can introduce this FISH probe to the denatured DNA, and it'll bind to the specific HER2 region of the cancerous DNA that encodes for the HER2. And by looking at this underneath a microscope, you can literally see where that HER2 gene is, which is really powerful. And note that FISH can be used not just for HER2 detection, but for a whole host of anything, as long as you just swap out the respective complementary DNA strand. So these are two photos from the paper that actually show the results of this FISH, or fluorescence in situ hybridization. On the left here, you have a HER2 positive case. So just to remind you, HER2 positive means that the HER2 gene is being overexpressed. There are too many of them. And you can literally see that for each of these cancerous cells, the blue splotches, you have a dozen or so of these bright red splotches. Those are those fluorophores that are glowing. And so in this HER2 positive case, you have more than 6 of these dots, which means it's HER2 positive. Compare that to the other end of the spectrum, and you have a HER2 negative case where there's only 1 to 2 splotches per cell. This represents that HER2 gene only being present in quantities around 1 or 2 per the DNA. So FISH is incredibly powerful and incredibly useful and incredibly accurate. However, it has its downsides. Namely, it takes about 2 days to run a FISH test from beginning to end and costs about $300 to $800. This is significantly more than a more popular alternative called immunohistochemistry, or IHC. IHC uses a fundamentally different procedure to detect HER2 overexpression, but the important thing to know about IHC is it's much faster, much cheaper, but importantly, it's qualitative. So IHC actually requires a doctor to look at the results of your IHC test and use their own judgment to figure out whether it's HER2 overexpressing or not. If you compare this to FISH, where you just need to count the number of dots, you can imagine that IHC is significantly more prone to errors. And in IHC, you have this huge range of so-called ambiguous cases where doctors are not able to make a judgment whether or not the cancerous cell is HER2 overexpressing. In these kind of ambiguous cases, doctors then need to turn to FISH to kind of elucidate the results. And so while FISH may not be kind of the most prominent HER2 overexpressing test that we have nowadays, it is still highly relevant for this kind of quantitative objective look at HER2 and backing up some of the results from IHC. So before I jump into the paper, does anyone have questions about microfluidics, FISH, or HER2 overexpression? Great. OK. So the paper that I'm looking at is specifically applying microfluidics, remember just tiny tubes and tiny volumes of liquid, to this FISH analysis and trying to make it more competitive. So basically, the objective of the paper was not to make the FISH test more sensitive or more accurate, because it already achieves that. The FISH test is highly accurate and incredibly good at determining HER2 overexpression. Instead, their goal was to reduce the time and cost of the FISH test, make it more accessible, and do that through microfluidics. So this is the actual microfluidic setup. This is super exciting, but it is a little bit complicated, so we're just going to focus on the left portion here. So these are 2 microfluidic chips, and you remember I mentioned these at the beginning. These are just where they build those tiny tubes. And each one of these microfluidic chips is 1/2 of like a clam that clamps together to form a microfluidic test chamber. So on the bottom here is a 2 centimeter by 2 centimeter square. It's about 5 microns deep, and this is the chamber where you load your cancerous tissue sample. So a researcher would take a sample from a breast cancer patient and load it into this chamber right here. And then they would take the opposing half of the microfluidic chip and clamp it on top, forming a sealed system. Now on the second half are two sets of like trees of these microtubes. And these microtubes are connected up to two inlet/outlets. These inlet/outlets are then connected out to very high precision pumps. And so what this allows researchers to do is basically introduce that FISH probe. And if you remember, the FISH probe, talked about that a couple slides back, is that little glowing piece that allows researchers to actually find the HER2 gene. And so they can introduce that FISH probe very precisely through these two kind of sets of microtubes. And so what this enables researchers to do is what's called a square wave oscillatory flow, a fancy way of saying they can pump the probe back and forth. So in a traditional kind of commercial FISH procedure, you just expose the cancerous tissue sample to this vat of FISH probe. You let it sit there overnight, and it hybridizes, and you come back in the morning and look at it under a microscope. But in this new microfluidic setup, they can actually pump it back and forth and very precisely control kind of how it behaves and interacts with the tissue sample. So this is a graph showing how they actually optimize their microfluidic setup. And we're not going to dive too deep into it because it has a lot going on. But what I want to focus on is this right region right here. So this shows the microfluidic FISH optimization of specifically the flow rate. So this is how much volume of that FISH probe is being pumped through those tiny channels every single second, measured in nanoliters per second. So along your x-axis, you have the volume of this FISH probe being pumped into that microfluidic chamber. And along the y-axis, you have the HER2 copies per cell. Just think of the y-axis as the number of glowing red dots per cancerous cell that we saw earlier. And generally speaking, the higher number of glowing dots per cell, the better the flow rate is, or better optimized it is. So in this case, they ended up finding that 1 nanoliter per second was the optimal flow rate, but that's not what I want to focus on. What I want to focus on is this 1,000 nanoliter per second flow rate. Because you'll notice that 100 nanoliters per second actually worked quite well. It had the second best result, second only to 1 nanoliter per second. Whereas 1,000 nanoliters was pretty terrible. In fact, it was so bad, it was completely unusable. So another way of saying this is 1,000 nanoliters per second does not work. The reason why I bring this up is because the difference between 100 nanoliters per second and 1,000 nanoliters per second is 900 nanoliters. That's 0.9 microliters of solution. These are incredibly small volumes, and yet the difference of 0.9 microliters per second is enough to take the test from almost perfectly optimized to completely unusable. This is why they need microfluidics. This drop right here is the explanation for why they need this complicated set of microtubes, because a 0.9 microliter difference in flow rate is the difference between a working test and a not working test. This is why they can't just shake the tissue sample around in the FISH probe to get that square wave oscillatory flow. And if you're wondering why that 0.9 microliter change in flow rate had such detrimental impacts, the extra liquid flowing through actually knocked off those FISH probes from the DNA. And so when researchers went to look at it under a microscope, they couldn't see the probes because they weren't there. They had been washed away. So the results. Now that they've tuned the system, now that they've built it, does it actually work? Well, overwhelmingly, yes. This is a graph showing the commercial FISH procedure as compared to the microfluidic-assisted FISH procedure. So along the x-axis is the number of those glowing red dots per cancerous cell as diagnosed by your bog standard FISH that you have in labs used nowadays. And along the y-axis is the same exact measure, but coming from those microfluidic tests that they just developed. And in a perfect world, we would hope that the microfluidic-assisted FISH performed exactly the same as the commercial FISH. In other words, if you were to give the same tissue sample to the microfluidic-assisted FISH and the commercial FISH, you would get the exact same number of red dots per cell. And that's exactly what we see here. We have incredibly strong positive correlation between the standard FISH and the microfluidic-assisted FISH, which shows that the microfluidic-assisted FISH performed incredibly well. So at this point, you might be wondering, well, why did we just develop this whole complicated microfluidic-assisted FISH if we just then proved that it performs exactly the same as a commercial FISH? What's even the point? Well, the point is that square wave oscillatory flow, that very precise control of the FISH probe back and forth through that microfluidic chamber, has amazing impacts on the amount of time and cost of the test. So a typical FISH procedure takes about 16 hours to hybridize. In other words, that FISH probe, the glowing little dots, they need to mix with your target DNA for 16 hours, which means it needs to be run overnight, and the entire FISH procedure lasts about 2 days, which is complex and costs money for lab workers. The microfluidic-assisted FISH only takes about 4 hours, which is a huge increase in efficiency and makes it more appealing for a doctor to prescribe it. Second, a microfluidic-assisted FISH is much cheaper. Because of the fine control that they have over the microfluidic-assisted FISH probe, in other words, how much of that volume of liquid they're actually pumping through the chamber, they're able to cut down on the amount of probe used, making the test a fraction of the original cost. So much so that it's now even competitive with IHC, that kind of other cheaper test we talked about earlier. So to summarize, microfluidic-assisted FISH performs almost, if not exactly as well as the commercial existing FISH procedure out there, but it's incredibly cost-effective and much faster. So as this goes from kind of a research paper into a commercial setting, which I certainly hope it does, it'll mean that people diagnosed with breast cancer and who need to get tested for HER2 overexpression now have this quantitative option, which is cheaper and faster and more accessible. So with that, I think that's the end of my presentation. Thank you so much for coming, and Anahita's presenting next week.